Hello, I'm Sophie Rodner from the American Chemical Society. Welcome to this media briefing from ACS Fall 2021. We're joined today by Dr. Thanh Do and Aleksandra Antevska from the University of Tennessee. They're studying how migraines protect against diabetes. Dr. Do.
Good morning, everyone. My name is Thanh Do, and my research group at the University of Tennessee is interested in unraveling the molecular connections between different diseases. So today I'm going to talk about our recent works on connecting these migraines and type 2 diabetes via probing the neuropeptide from single mouse islet. So this project, we emphasize on not only the molecular connection of the disease, but on the new analytical workflow that we optimize and develop to samples, volume limited samples. So let's talk about the two diseases. We have migraine, which is the disease that occur in our head, right? When you feel migraines, it is the form of headache. And as a chemist, we characterize migraines by the neuropeptides that elevate under these conditions. And there are two set of peptides that we are interested in. The first one is calcitonin genes related peptides, or Cgrps. And then the second one is pituitary amylase silase activating polypeptides, or Picaps. So in the migraine patient, whenever they feel pains, these peptide concentration would elevate. They bind to the receptors and triggering the pain feeling that we have. Now, talking about diabetes, when you think about it, is a disease that happens in the pancreas. Specifically, if you look at the cartoon figures of the pancreas, you can see that there are spherical structures that color in orange. So each of those is an islet of Langerhans. And each of the islet, they contain a cell cluster. So there are four major cell types: alpha, beta, gamma, and delta. So the beta cells, they produce insulin. And as we all know, insulins regulate the blood glucose levels. So in this study, we connect seemingly different disease, migraine and type 2 diabetes, and we try to explain why migraines may show certain protective effects against type 2 diabetes. Next slide. So if we look at the diabetes in general, we often do not study the disease using human subject. We use animal models, and in this case, we use either mouse or rats. So rodents in general is the most commonly used animal models in diabetes research. However, for mouse, they have two different genes encoding their insulin, and we often call them insulin 1 and insulin 2, while in humans, we only have one version of insulin. So right there, it poses a challenge on determine whether we can not only measure insulin in mouse, but being able to distinguish between insulin 1 and insulin 2. So we work in collaboration with Professor Michael Kalstad in Department of Surgery at UT Health Science Center, and he is the leading expert in isolating healthy mouse islet. And then we use it in our LC ion mobility mass spectrometry workflow. And the goal of this study is being able to quantify the level of insulin 1 and insulin 2 under different conditions. So one example you can see on the left screen is a mass spectrums. And as for you, you can see just a lot of peaks. But for us, based on the sequence of the protein, we can rely on the mass spectrum to determine the levels of insulin 1 and insulin 2 in these islet sample. So one thing I want to emphasize is that here we make a measurement at the single islet levels. So each pancreas can give us about 100 to 200 islets, and that means we can perform 100 to 200 experiments on a single mouse, and that would save a lot of animal costs and also allows us to do a lot more experiments. Next slide. So in our group, we have a very unique techniques called eye mobility mass spectrometry. The idea is that with traditional mass spectrometry, you can only measure the mass to charge of the molecules. So if the molecule has multiple conformations or form several oligomers, you would not be able to see those because they often consider as isobaric species. But with eye mobility, for example, you can see in the lab panels here, we can separate insulin 1 and insulin 2 and then look into their conformations. You don't need to know much about eye mobility to see that insulin 1 and insulin 2 has distinctively different structures. And on the right panels, we compare the insulin levels under different treatment conditions. Let's first focus on insulin 1. So insulin 1 is considered the defective form of insulin in mouse. It has been shown that mouse contain only insulin 1 would develop diabetes very early in their age. So what you need to remember here is that insulin 1 is bad. So if we produce a lot of insulin 1, that can accelerate the diabetes conditions. And as you can see here, under 4 condition, the control, where we have no peptide added to the system, or where we incubate the peptide with amylin. Amylin is a peptide that has been implicated in the development of type 2 diabetes. And then Cgrps and the mixture of amylin and Cgrp, you can see that in all of these peptide treatment conditions, the fresh peptides seem to suppress the level of insulin 1. However, in the case of the ages peptide, Cgrp significantly stimulate the level of insulin 1. On the other hand, insulin 2 is considered the human orthodox. So it's behaved like our human insulin. It's a good version of insulin. And as you can see for all these peptide treatment, Cgrps or amylin or the mixture of the 2 peptide, the insulin 2 level was suppressed. So what we believe here is that in human, we don't have the insulin 1 version, so we don't have to worry about this defective form of insulin. However, for insulin 2, the Cgrp play a role in regulating the amount of insulin. And as you can see, if we release insulin in the minimum amounts, that would protect us from insulin resistant and later on develop diabetes. So from this study, we believe that Cgrp plays a critical role in diabetes protection. And for the first time, we was able to show the protective effects the dual effect of Cgrps on insulin production in rodents.
Thank you, Dr. Do and Alex. Now I'd like to ask you a few questions about your work. First of all, can you tell me, starting from the beginning, what are neuropeptides?
So neuropeptides are small proteinaceous substances that are produced and released by neurons.
And what is their role in the body?
So they're the most abundant chemical messengers in the brain, and so their most prominent role is to modulate. So it's to basically they're like key mediators between the communication between neurons and like those muscles, the immune cells, and all that.
So one way you can think about neuropeptides is, let's say I am a cell and I want to communicate with other cells. How I can do that? Well, I can do it by produce very little peptides that may just last about 5 or 10 minutes in vivo, but then it's going to bind to a receptor on a cell surface. And then by binding to the receptor on a cell surface, it triggers activities in the cells, either open up an ion channels or changing the production of certain peptides or protein or respond to the lipids. So the neuropeptides are very important and is abundant in our body. It can produce by many cells, but most of them are produced by neurons. So that's why we have a term, neuropeptides.
What got you interested in studying them?
For me, it's back in my postdoc where I work with Professor Jonathan Swiller, who is an expert in finding, characterizing, and connecting the structure and function of neuropeptides. And in this case, in this particular case of migraine and diabetes, as a chemist, we characterize the disease not by the symptoms, not by how long the pain is, or how the patient feel, but we characterize the disease by the chemicals. And in this case, the neuropeptide is what we look at. Of course, it's just one way to do that. You can characterize the disease by a change in lipids or change in protein. But neuropeptide is just cool because the molecules are always acting on the system, right? And so if we determine their function and structures, and we can see how they connect, how they have different function in different organs, and potentially we can connect disease that way.
So how are migraines and diabetes related?
It first is a surprise, right? A lot of time you would hear that, getting one disease would increase the risk of having another disease. Like a lot of time people say, well, if you have diabetes, then you would have a higher chance of getting, cardiovascular disease, or you may have increased risk of having Alzheimer's. So we all heard that. But what happened about the disease? Of course, getting migraine is not something that people enjoy, right? But having migraine somehow protect against diabetes. And we find out by looking at the literatures, and then there are two big studies. One study happened in France, where they look at a very large population of women, about 74,000 women, and then they followed their medical history for a long time, and then they discovered that those who have migraine tend to have a lower risk of diabetes. And let me explain that statistic a little bit. As I understand, it's not, out of those 74,000 women, there's a subset of those people who may have diabetes, and then there's a subset who may have migraines. And so they look at the subset who have diabetes and migraine, and they discover that, for the people who have migraine, the risk of having diabetes decreased from 22% down to about 11%. Okay, so of course, that is just a study that report the statistic. And as a chemist or a, try to be a biologist here, we try to use our tools and try to understand what would cause the protective effect. Yeah.
What do the peptides Cgrp and Picap do?
So Cgrps, or calcitonin genes related peptides, it's actually a series of peptides that have very similar sequence, but they're different by a few amino acids. And so I often refer to Cgrps as pain peptide. Okay, so if you somehow hit your fingers with a hammer, right? When you work and you hit your finger with a hammer, you feel pain immediately. It's because your neurons, mostly your dorsal root ganglion neurons, they release the chemicals, and most of the time it's Cgrps. And Cgrp, like I said, would bind to receptors and triggers either a dilation of blood vessels or a feeling pain. We feel pains because something happened to our body. It's actually the chemical that causes pain. Instead of, a lot of times people think pain has to be physical, but in this case, pain is chemical, right? And same thing with Picaps. Picaps is another type of neuropeptides. And interestingly, the Cgrps and the Picap, they have very similar sequence length. So I think the Cgrp is about 37 residues, so made of 37 amino acids, and then the Picaps is 38 residues, so 38 amino acids.
And what is the role of insulin in the body?
So of course, I think all of us heard of insulin before, right? Insulin would help us regulate the glucose level in our blood, right? When we have a big meals, probably all of those fat and protein and carbohydrate would turn into glucose. And then there has to be a hormones. We like to think insulin as a hormones that would regulate the level of insulin by, letting the fat cells, or those cells that would absorb the glucose, right? So, but then the more we learn about insulin, it's actually a lot more complicated, right? So if you think about the insulin available for diabetes people, there are actually 6 or 7 types of insulins. So there's those that is fast acting. So when you get those, it, immediately, regulate your glucose blood levels. And then there would be something that is long acting or slow acting. It really depends on, the goal of either you want to have a immediate effect, or you want the effect to last really long. And so even though you don't really ask me, you have not asked me the question, but we tend to think that there are different kinds of insulin. Not all insulin are the same, especially in the context of rodents. And by rodent, I mean mouse or rat. They have 2 different insulin. They may behave very differently. And we think that it's important to be able to distinguish and understand the difference.
And what did you discover about the effect of the neuropeptides on insulin in rodents?
So for this one, we actually found out that the neuropeptides such as Cgrp has actually a dual effect on the insulin on the mouse 2-gene insulin system, and Dr. Do mentioned previously briefly about this. But we found out that the Cgrp actually stimulates the levels of insulin 1, and then it suppresses the levels of insulin 2. So it has like a different effect on the 2 insulin gene system.
So one thing I have to add is, people have been studying mouse for a long time. They know that insulin 1 and insulin 2 exist. They even know that insulin 1, they can generate transgenic mouse model where, the mice only have insulin 1, and they show that it's accelerate the diabetes development. And in other transgenic mice where they change the genes and make the mice produce human insulin, in other words, insulin 2, they see that it's protect the mice from getting insulin. But then when you see those data, you would ask yourself, well, it's just insulin, why they have different behaviors? And in our study, we actually discovered something really cool. The Cgrp peptide, human has Cgrp peptide, mouse has Cgrp peptides. Cgrp peptides stimulate insulin 1. And remember, if insulin 1 is the bad insulin, if the mice only have insulin 1, Cgrp stimulate the production of insulin 1. Soon enough, the mice will develop insulin insensitivity, right? Will lower the level of sensitivity to insulin, and the mice going to develop diabetes. On the other hand, human, we don't have that bad insulin, we just have the good insulin. So if Cgrp helps regulate of that insulin 2, that actually make the insulin more sensitive, right? And we tend to think about the beta cell as the machine that produces insulin. If you make the machine work day and night, produce a large amount of insulin, soon enough they're gonna experience wear and tears and die, right? But if we just make it work maybe 4 hours a day and enjoy the rest, then potentially we have a long-lasting beta cells that protect us from getting diabetes.
Now, diabetes involves dysregulation not only of insulin, but also of amylin. What is amylin and what is its role in the body?
So amylin is a neuropeptide, not a neuropeptide, it's a peptide that was co-secret with insulin from the beta cells. So for a long time, people did not know about the function of insulin, a function of amylin. recently they suggest that it may have certain function in, generating gastric acid. And we tend to think about amylin as like a checkpoint on the production of insulin. So let's say if your beta cell produce a lot of insulin, how do the cell know when to stop? And probably if you use amylin, that's say like, okay, that's enough. Let's stop the production. Now, the reason people were so interested in amylin because human amylin can aggregate to form fibrils. So this is very interesting. So when you think about a peptide, you think about a long chain of like a polymers, right? And when we talk about aggregation, certainly these polymers, they can clump together and they clump into a specific way that lead to a micron long structures like our hair, but under the microscope, right? So this is a fibril structures. And that only happened with human amylin and not rodent amylins. So most of the time you do not hear people talk about mouse, like regular mouse having type 2 diabetes. It's because people think that because the rodent amylin does not aggregate and only the human amylin aggregate. So most of the mouse models used in type 2 diabetes research are transgenic mouse model. That means they have to change certain thing in the gene so that the mouse can produce either human amylin or something else that's gonna generate the symptoms of diabetes. So for a long time, people interested in this amylin. And so in the healthy person, the ratio between insulin and amylin is probably, I would say 1,000 to 1. So a lot of insulin and very little amylin. But in the type 2 diabetes patient, the ratio is increased. So you may think about 100 to 1 or even 10 to 1. So you have a lot more amylin. And that makes sense, right? Because type 2 diabetes people tend to have, high blood glucose. They need constantly produce insulin to keep the glucose level in check. And then these amylin, when they accumulate, right? When they have a right enough concentration, they actually accumulate. And during that process of forming the fibril structure, we think that they form certain toxic oligomers that may kill the beta cells. Right? The beta cell produce amylin, and then the amylin oligomers then kill the beta cells.
All right. Now, given the positive effects of the neuropeptides, would it be feasible to use them to treat someone with diabetes?
Yes, I think, of course, we do not want to use the native form of Cgrps or Picaps in the treatment of diabetes because we know for sure that they may have different functions in different organs. And one example is migraine, right? The migraine is characterized by elevated concentration of Cgrps. But we can look into the analogs, or we can look into maybe the truncated form of the peptides and see if we keep chopping the peptide to a shorter and shorter version or make chemical modification, which chemists is very good at, then we can come up with analogs that just do the specific function that we want. And the nice thing about Cgrps is we actually have two different cases. So in one case, the Cgrp oligomers appear to stimulate insulin 1. Now, we said stimulate insulin 1 is not a good thing, but how about stimulate insulin 2, right? So if we can develop, analogs based on Cgrp oligomers that stimulate insulin 2, that's a good thing too, because a lot of time the patient, they need to produce insulin, right? They need to have some form of insulin because their beta cells no longer working. In the other case, we have Cgrp suppressing or regulating the insulin. So having an analog would be nice, would be an alternative approach to curing this disease.
Thank you. Media briefings for ACS Fall 2021 will be posted throughout the meeting at www.acs.org/ACS Fall 2021 briefings.